Good day, students. Till now we have discussed the overall structure of the cell and the various cell organelles like single membrane bound organelles, double membrane bound organelles, organelles without membrane, and the general structure of cell, cytoskeleton, etc. We have also studied the differences between animal and plant cell and the eukaryotic and prokaryotic cell. Now, in today's lecture, we are going to discuss about some important points regarding the cell. Now, the first point is the centrosomes. As you might be knowing till now, or you are familiar with the word centrosome or centriole, you must be knowing that the centrosomes or the centriole are present only in the animal cell. So, first point is regarding the centrosomes. As I told you earlier, that the centrosomes are present only in the animal cell. So we should write found only in the animal cell. And if we talk in the terms of level of organization, that is prokaryotic or eukaryotic, then we should say that the centrosomes are found only in the eukaryotic cells. If we talk about the structure of a centrosome, it consists of two centrioles. Centrioles or the centrosomes are composed of the microtubules. They are made up of. We have studied the three types of filaments that make up the cytoskeleton, that is microfilaments, microtubules and the intermediate filaments. So it is the microtubules that make up the centrosome. Now coming to their function, what is their function? They help in the spindle formation during cell division. So they are very important. They play a very important role during the separation of chromosomes during cell division. So they help in cell division by forming the spindle fibers. We should also keep in mind that the centrosomes are membrane-less structures. After knowing about the centrosomes, let us discuss one more structure, that is the cilia and flagella. You must have heard these terms, cilia and flagella, and these are the structures that are related with the movement and locomotion of the cell. So, let us discuss cilia and flagella. Cilia and flagella are thread-like structures that arise from a structure that is called as basal body. These structures, that is cilia and flagella, help in the locomotion. You all understand locomotion? Locomotion means movement of the cell. They help in the cell movement from one place to the other. Now the question arises, what is the difference between cilia and flagella? Okay, before talking about the difference between cilia and flagella, one more important point is that they are made up of the microtubules. The main difference between cilia and flagella is that the cilia are numerous and smaller structures present on the cell, whereas flagella are longer whip-like structures, and in a single cell their number can vary from 1 to few. So one difference is that cilia are numerous, whereas flagella are few in number, or they can be one in number, and cilia are smaller in their size, whereas flagella are longer and whip-like structures. For example, if you have seen a paramecium, which is a protozoan, throughout its surface small structures are present that are called as cilia. And if we talk about euglena, you can clearly visualize a single structure that arises from one of its ends, and that is called as flagella. Now, after knowing about cilia and flagella, let us discuss one more structure which is very important, and that is called as chromosomes. These are the heredity material. They contain the heredity information in form of genes. So chromosomes are the heredity vehicle. Heredity means the transmission of characters from one generation to other. So they are the vehicles which transmits the characters from the parents to the offspring. So we can say them as the heredity vehicles. Heredity is related with the transmission of characters from one generation to other. If we talk about the structure of a chromosome, when the cell is in resting stage, and that means when the cell is not dividing, the chromosome lies in the form of a thread-like intermingled structure, a structure in which you can visualize where the threads are intermingled among themselves. So chromosomes lies in that form during the non-dividing phase, but when the cell is in dividing phase, when it is ready to divide, then these thread-like structures get condensed and take a particular shape and structure, and that is called as chromosome. When these chromosomes lie as a thread-like structure, that is called as chromatin network. So we can say they are thread-like structures. Let me remind you once again that these thread-like structures are called as the chromatin material, and it occurs in the non-dividing cell, whereas these thread-like structures get compacted and condensed when the cell is ready to divide, and they take a particular shape and structure that is called as a particular chromosome. Both can be called as the chromosome invariably. Now, you should ask one question: where these chromosomes occur. Now, after you have studied the whole structure of cell, you can easily answer this question. If we talk about a eukaryotic cell, the chromosomes lie inside the nucleus. But if we talk about a prokaryotic cell like bacteria, where does this chromosome occurs? It occurs without any membrane like covering, that is, it does not occur in the nucleus, but it lies in the center of the cell, and that is called as the nucleoid. So in prokaryote there is only one chromosome, which is circular loop-like structure which occurs in the nucleoid, whereas in case of eukaryotes there are numerous chromosomes that occur in the nucleus. Now After this chromosome, we have studied the membrane also, which forms the covering of a cell, which separates the internal environment of the cell from the external environment. And now we are going to study some terms regarding that membrane, and that is the permeability. Permeability means the capacity of the membrane or the property of the membrane to allow certain things to pass across it. So, on the basis of what materials it can allow, we divide the membrane into three categories. So, now we are going to discuss about the permeability of the membrane. We have divided the membrane into three types on the basis of their permeability or property. What is permeability? Permeability means the capacity or the property of the membrane to allow the things to pass across it. Now, what things it allows to pass across is decides the permeability property. Now, on that basis, the first type is permeable means the membrane which allows the diffusion of both solvent and solute molecules through them. Example of it is cell wall. Also, we have discussed this property when we were studying the cell wall, that cell wall of plant cells is permeable, which allows the diffusion of solvent as well as all the solute molecules through them. Now, the second type of membrane is the impermeable. It is opposite to that of permeable. So, we can clearly understand what is impermeable. It means the membrane which does not allows the entry of either the solute or the solvent molecule through them. Now coming to the example of impermeable membrane, the lignified and the cutinized cell wall of the plant cells become impermeable to anything. That is, they don't allow either the solute or the solvent molecule. The third type of the cell membrane is the semi-permeable membrane. Membrane which allows only the entry or passage of the solvent molecules but do not allow the passage of solute molecules through it, then it is called as a semi-permeable membrane. This type of membrane allows diffusion of solvent molecule only. But it does not allow the passage of the solute molecules. Now example, the membrane surrounding the kidney is the semi-permeable membrane. Semi means which is allowing the diffusion of solvent only. So example is kidney. The last type is the differentially permeable. Differentially permeable membrane is that membrane which allows the diffusion of solvent molecule, but beside that it also allows the diffusion of some selected solute molecules. So it allows the diffusion of solvent plus selected solute molecules. Example of differentially permeable membrane is the plasma membrane of the cell, that is called as plasma lemma, and tonoplast. You must be remembering tonoplast is the membrane which surrounds the organelle, that is called as vacuole. So examples are plasma lemma, that is plasma membrane of the cell, and tonoplast, which is the membrane of vacuole. So in summary, we can say there are four types of membrane on the basis of their permeability. Now, let us discuss some more important points. One such point is, one such structure is basically plasmodesmata that we have to discuss. Plasmodesmata is the structure which connects two cells, and it allows the communication between two cells, and it is present mainly in the plant cells. It can be defined as the connection between two neighboring plants. These plasmodesmata allows the chemical connection or chemical communication between two cells. If we say, for example, this is one plant cell depicted by the red color, and this is the this is the second cell wall, second cell, neighboring cell, adjoining cell, and this is the cell wall of the second cell. Then there is a connection between the two cells through which they can easily communicate. These are two neighboring cells. The red color is depicting the cell wall. This structure, this connection is called as the plasmodesmata. Although it is not continuously opening, it is also guarded by some structures, but it provides a connectivity between the two neighboring cells. Guard the opening of the plasmodesmata, but it provides a certain level of communication or connectivity between the two cells. Now, after plasmodesmata, the next point is protoplasm. We have studied in brief about the protoplast. The protoplast can be called as the physical basis of life. And this statement, that is, the protoplast is the physical basis of life, was given by Huxley, the scientist. And what does this statement, that is, protoplasm is the, sorry, it is protoplasm, not protoplast. This statement means that protoplasm is the site where the chemical energy is converted into biological energy. Say for example, chemical energy is present in the bonds of the chemical compound like glucose, and it is converted into the biological energy, that is in the formation of ATP and other substances which are essential for the functioning of cell. So the protoplasm can convert the chemical energy into biological energy. There is one more important fact, that euglena, which is a protozoan, is the connecting link between the plants and animals. Euglena is a unicellular protozoan which has a characteristic feature of an animal cell, and it is eukaryotic. One more important fact is that the mesosomes. Mesosomes are the structures which are found in the bacteria, which are prokaryotic organisms. Mesosomes is analogous to the mitochondria, since they help in the cellular respiration because they contain the enzymes which participate in the respiration reaction. And mesosome is nothing but the infolding of the plasma membrane of the bacterial cell. So, you can say mesosomes of bacteria is analogous, means similar in function So, mesosomes is analogous to mitochondria, means it is similar in function to mitochondria because mesosomes contain the respiratory enzymes, but the structure of mesosome is entirely different from that of mitochondria. In case of plant cells, we know that they contain a covering that is called as cell wall, but in case of animal cell, they have cell coat instead of cell wall, and it is made up of gliocalyx. The function of cell coat is firstly to provide protection just like cell wall, and besides that it helps in cell-cell recognition. Now question arises, what is glycocalyx? Glycocalyx is nothing but it is the glycoprotein and glycolipid part of the plasma membrane. We have studied it in the structure of, when we were studying the structure of cell membrane, that the membrane lipids and membrane proteins on the outer side of the cell towards the external environment contain the carbohydrate chains attached to them. So they are called as glycoproteins when the carbohydrate chain is attached to protein, and glycolipid when the carbohydrate chain is attached to the lipid particle. Should I repeat it again? That glycocalyx means the carbohydrate chains which are attached to the lipid and protein parts of the membrane. Glycolipid means carbohydrate attached to the lipid part, and glycoproteins means carbohydrate attached to the protein part of the membrane. And these glycocalyx help in the formation of cell coat, which helps in cell protection as well as cell recognition. Next point is that about ATP. ATP is also called as adenosine triphosphate. It is the energy currency of the cell because the cells can utilize energy only in the form of ATP. As the protoplast is called the physical basis of life, DNA is called the chemical basis of life. DNA stands for deoxyribose nucleic acid. DNA makes up the chromosome. Chromosome we have just discussed in this lecture also, that chromosomes contain the genes, and these genes are made up of, chemically made up of DNA, and DNA is deoxyribose nucleic acid. It is a macromolecule or a polymer made up of nucleotides, and it forms the chemical basis of life because this is the chemical which has the information for transmission of characteristic from one generation to another generation, as well as for control of the cells all functions. So it is the chemical basis of life because it is the controlling molecule which control all the functions of cells as well as the transmission of characters. And the structure of DNA was proposed by Watson and Crick, and according to them, the DNA consists of a double helix. Double helix model explains the structure of the DNA. Last point. We have studied when we were studying the structure of mitochondria and plastids that they have their own DNA molecule. Besides that, centrioles or the centrosomes also have their own DNA molecule. So therefore, they are called as semi-autonomous cell organelles. So, mitochondria, plastids, and centrioles are called as semi-autonomous cell organelles. Since they have their own DNA molecule. This means they can synthesize some of their proteins independently without the help of nucleus, but for other proteins they are dependent on the nucleus. Therefore, they are called as half dependent, half autonomous. That is, they can make some of their own proteins. So, this is all about today's lecture. Have a good day.